So today what we're going to be talking about are the different types of microscopes. The microscope is an extremely important tool when it comes to the science of biology because a lot of what we study in biology is smaller than what the eye can see. So if you look at the word microscope, it breaks down into two Latin root words, micro, which means small, and then scope, which means to see or to look. So literally a microscope is something that helps us to see small things. And many of you have probably used microscopes before, and the picture on the right probably is most recognizable as to what you think a microscope looks like. But ancient microscopes that were invented hundreds of years ago, operated slightly differently. Little object in the upper left-hand corner, this was one of the original microscopes invented by a scientist, and it was this kind of small piece of brass about this big, and you would hold it up to your eye and look through the little hole that's right there in the middle, and then you would put a sample on the end of the needle and use the little screw knobs to adjust your sample, and then you would be able to magnify things. So anything that can make a small object appear larger could be considered a microscope. A magnifying glass is a simple microscope in that it has a lens, it makes things look bigger so you can see things that are smaller than the eye can see. Okay, so the microscopes that we use today are more sophisticated and allow us to see a lot more. But the invention of the microscope actually gave us a whole new view on the world and just life in general. Prior to the invention of the microscope, people had some very interesting ideas about how life worked. Okay, someone would go down to a lake or a pond or something and scoop up a bucket of water to cook their dinner, but this would be the same lake or the same pond that someone would be washing their laundry in or bathing their cow in, and they didn't understand why people were getting sick. When they invented the microscope, people were finally able to take samples of that water and see, oh wait, there's tiny little things running around inside of that water. Okay, maybe drinking that is not good for me. Prior to understanding that there were microscopic germs, people used to think, oh, you get sick because you're a bad person, and this is, life or God or whatever punishing you for being bad. Or it was a full moon out, so people get sick on the full moon. Okay, we had no idea. They used to think that you would have evil spirits in your head and we could drill a hole in your head to let the evil spirits out. Yeah, that will cure your illness. Okay. So once the microscope was invented and we can finally see these tiny organisms that were making us sick, it changed our view of the world, it changed our view of science, and also changed our view of medicine for the better, I would say. Okay, so the microscope is an extremely important invention in terms of allowing biologists to do their work. And a little bit later on, we're going to talk about some of the biologists that were influential in kind of developing our ideas of life science, specifically in our understanding of cells. And the invention of the microscope was critical to their work. So today we're going to look at kind of the different kinds of microscopes that there are. And the microscope is a tool like anything else, and each kind of microscope is useful for a different type of job. So depending on what you want to accomplish with the microscope determines the type of microscope you'd want to use. So the first one we have here is the compound light microscope, or CLM for short. And this is the most common type of microscope used. We'll see it in high schools, we see it in college, we see it in professional research facilities, we see it in forensic crime labs. So this is the most common type of microscope out there for a couple of reasons. First of all, it's relatively inexpensive. If you look at the price range, a working science grade microscope can be anywhere from $100 to about $5,000. Yes, I know you can get a really cheap microscope from Walmart or something, like a kid microscope. Those are technically microscopes, but you wouldn't use it for any serious sort of scientific investigation. So something that you could use on a professional level falls within that price range. Now, you might say $5,000, that's not cheap. Well, compared to some of the other microscopes we're going to see today, yeah, that is on the less expensive end of the scale. But the other reason why these are very common types of microscopes is because they're relatively easy to use. Okay? Every year I teach a classroom full of students how to use these microscopes in about 20 minutes. Okay? Like anything, it takes practice to get good at it, but I can show you the basic workings of a compound light microscope in relatively short amount of time. I mean, I've done this for middle school classrooms where I've had groups of like squirrely 8th graders and I've taught them how to properly use a compound light microscope. So it's relatively simple and it's relatively readily available to kind of the general public. So these are very, very common microscopes. And we're going to go over the parts of the microscope and how it works in more detail when we actually work on a lab using these microscopes. The simple way that these work is at the top you have the eyepieces. You look through those eyepieces, and then in the middle you have these objectives. These objectives are the lenses that we can change and change the magnification factor of that microscope. And then at the base, you're going to have a light source of some sort, whether it's a regular incandescent light bulb or an LED light bulb. Okay, what happens is the light is going to shine through the sample that we put on the platform, the stage. So light is going to pass through the sample and then up through the lenses and get magnified and eventually reach your eyes. So what you're looking at is a magnified version of the sample that you have on your stage. A very important component of the compound light microscope is that the light has to be able to pass through your sample. So only very thin samples can actually be viewed underneath a compound light microscope, something thin enough where light can pass through. Oftentimes when we get out the microscopes in class, I'll have some knucklehead go, "Can I look at my fingers under there?" And they stick their fingers underneath the microscope, and to their shock and amazement, they can't see their fingers. Well, that's because your fingers are too thick for the light to pass through, so you're not going to be able to image something there. It has to be a relatively thin sample. Light has to be able to pass through to get to the lenses to get magnified. Another interesting advantage of the compound light microscope is that you can actually look at living specimen underneath the microscope, assuming those living specimen are relatively small. So here we have a picture from a typical CLM. These are single-celled organism called paramecium. Paramecium are a very common type of microorganisms that you would find in like a lake or a pond. So if you go down to like Lake Hollingsworth or Lake Mirror or someplace like that and take a scoop of water out of there, there's a good chance you can find paramecium in there. Okay. Now, since the light is passing through the sample, what we're going to see here is the inside of those paramecium. So we're seeing the outside boundary of the paramecium and the little hairs that surround them that helps them swim around in their environment. And then we're seeing all the little bits and pieces inside those cells. A typical CLM gives you a magnification factor somewhere between 40 to potentially 5,000 times magnification. That is, whatever you're looking at underneath the microscope, it will be 5,000 times larger than normal. And that's a pretty top-of-the-line type of CLM. The ones that we have in class go up to 1,000 times magnification, but the 1,000-time magnification objective is what is called an oil immersion lens and takes a special technique to work properly, so we don't typically worry about that one. The next type of microscope is what is called a confocal microscope, or CFM for short. So this is basically a modern spin on the compound light microscope. The compound light microscope more or less works the same way today as it did hundreds of years ago. You shine light through a sample, up through a series of lenses, and you get a magnified image. Okay. Now the lenses have improved, and now we have like digital cameras and computers that we can attach to these things to make them much more useful, but they more or less operate the same way. So about 40 years ago, scientists said, well, what if we take this same concept of the compound light microscope, but try using a more intense light source? So what if we use a more focused light source like a laser? Not only can we use one laser, we can use multiple lasers, because lasers are very thin, so you can shine multiple lasers through a sample at the same time. And this is the confocal microscope. Okay, the word focal meaning we're focusing several lasers on our sample at the same time. Now, you can't just turn on a laser and look through a lens and get an image, because that laser would burn your eyeballs out. So you can't do it that way. So what happens is we use the lasers to shine through your sample and then use sensors on the other side to pick up the information and send that information to a computer. The computer then processes the information and turns it into an image. The advantage of a confocal microscope is that since we're shooting multiple lasers, we can change the angles of those lasers, and we can actually produce a three-dimensional image as a result. A CLM only produces a flat two-dimensional image. Here we can get both two-dimensional and three-dimensional images. Now, someone might observe when you look at this microscope is, oh, it has eyepieces there. I thought we couldn't look through it. Well, this is kind of a two-in-one microscope. It has a normal light source, so you can use it like a normal CLM, but then it also has the laser light source and the sensors that connects to a computer. The price range here is a little bit steeper, so anywhere between $100,000 to $750,000, depending on the sensitivity of the sensors and the intensity of the light being used. So you don't typically see these in like a high school classroom because they're just far too expensive. They're a little bit harder to operate. It would take a little bit longer to get good at imaging these things. So you'll typically only see these in like professional research labs. So here's an image from a CFM. This is actually looking at part of the cochlea inside of your ear. So inside of your ears, you have a small organ which kind of looks like a snail shell. And inside this snail shell organ, there is fluid. And within this fluid are tiny little hairs. Okay? And that's what we're looking at here. Now, the image that you see has multiple colors on it. That's because when you send that information to the computer colorizes it for you. Okay, the lasers don't have specific color to them, but the computer basically assigns color to the image so it's easier to see. Because our eyes process color much better than they process like black and white images. So it's colorized. This is what is known as false color, because this is not what really the hairs look like inside the cochlea. What these hairs do is they actually help you maintain balance, because the fluid inside of that little snail-shaped organ sloshes around whenever you tilt your head. So if you tilt your head to the side, the fluid moves and those hairs kind of move back and forth. And the hairs are actually connected to nerve endings. And when you tilt your head, what happens is the cochlea sends a signal to your brain that says, hey, we're sideways. Okay, so that way you can kind of determine your orientation. If you've ever spun around in a circle and then stopped suddenly, like even though your feet stop moving, it feels like you're still moving and your head is still spinning. And that's because basically you're swirling the fluid in the cochlea, and when you stop, the fluid still swirls for a little bit. So it's sending a signal to your brain that, hey, we're still moving, but your feet is sending a signal saying, no, we're not. Okay, and that's where you get that kind of like dizzy, almost sick to your stomach feeling, is because your brain is receiving different messages from different parts of your body. This is also what contributes to like car sickness and air sickness and seasickness, is when you feel your body moving, but you don't really see it moving, especially when you're like in a car. If you're like looking at like the dashboard or the person sitting next to you in the driver's seat, okay, if you're sitting in the passenger seat, like your body feels that you're moving, but your eyes don't see that movement unless you're sitting there staring out the window. So it's that disconnect between what your different parts of your body are sensing. Okay, that's what causes that like nauseous feeling of car sickness or seasickness. And that comes back down to the hairs in your cochlea. All right, so here's the first formative question. Why wouldn't a CLM or a CFM be useful for looking at your own fingerprints? I've kind of alluded to this earlier. A, the light from the microscope would burn your hand. B, the microscopes cannot look at things that are large. C, your fingers cannot fit under the microscope objective. Or D, the light from the microscope cannot pass through your fingers. The next type of microscope is what scientists thought of is, well, what if we, instead of using light to image something, used another source of energy? So we have what we call the electron microscope. And there's two different types of electron microscopes. This is the first kind, called a transmitting electron microscope, or sometimes called a transmission electron microscope. Okay, so this works very similar to the CLM, that we're going to shoot a beam of energy through a very thin sample. But instead of shooting a beam of light and then processing that light with lenses, we're going to shoot a beam of electrons. So a beam of electrons is essentially electricity. So we're going to shoot a beam of electricity, a beam of electrons through the sample. And our eyes can't see electrons, so we have to have computers to process the image. Okay, so essentially what happens is the beam of electrons that's passing through the sample get bounced around. And there's sensors on the other side of that sample that picks up where the electrons bounce to. Kind of imagine if you had someone standing against a wall, and then you get a bunch of paintball guns, and you start shooting paintballs at this person. Okay, some of those paintballs are going to miss them, go whizzing right by their head, hit the wall behind them. Some of them are going to hit them square. Some of them are going to hit the side and like kind of ricochet. So if that person like steps away from the wall, you're going to get like an outline, an image of what that person looked like because of all the paintballs that went past them. Okay, now if you can imagine that on a smaller scale, and those paintballs can actually pass through an object, anything on the inside of that object is going to cause those paintballs to bounce and ricochet as well. So this is what's happening, but with electrons, is the electrons are hitting the inside of that sample and bouncing around, and the sensors on the other side pick up where those electrons land and send that information to a computer. Okay. Now, to be clear, in this image right here, this whole apparatus is the microscope. So this is a big piece of equipment, and it has a price tag to match. So anywhere between $85,000 to $500,000, you could get a couple nice cars for that amount of money. So there's a reason why McKeel doesn't have any of these lying around. The other reason why electron microscopes are not commonly found in high schools is because they are fairly difficult to operate. To prepare the sample to put underneath the microscope, you have to basically encase a sample in a type of plastic called acrylic and make very, very thin slices. This takes some time and effort to do. I went to the University of Florida, and they had a graduate level course for how to operate one of these kinds of microscopes. So you had to spend a semester learning and practicing how to use this microscope to operate it effectively. Now, if you had a sample that you wanted to look in very close detail, there are companies that their whole job is to operate electron microscopes. So you could send them your samples and say, hey, I want these images, and they will process the samples, get the images, upload it on a flash drive, and send it back to you. The magnification factors that you can get from a transmitting electron microscope can be 100,000 times magnification. So a typical CLM can go up to about 5,000 times. A CFM, confocal microscope, can go up to like 20,000 times. Here we're going up to like 100,000 time magnification. So what you're looking at is 100,000 times larger than normal. So the image that we have here is a mitochondria. This is a piece that's inside of the cell. So if you think about the paramecium that we saw in the first picture with the CLM, that's the whole cell. So we're zooming in even closer to see little pieces inside of the cell. When we first discovered cells, we didn't understand their inner workings. So it took instruments like the transmitting electron microscope to allow us to see, well, what parts are going on inside of the cell that makes it work. Okay. And we have a much better understanding of cells today because these instruments allow us to get a much closer look at their inner workings. Now, another kind of electron microscope that exists is what is called the scanning electron microscope. And this works in a similar way in that we're going to shoot a beam of electrons at our sample, but instead of shooting the beam through your sample, we're going to bounce those electrons off the surface. Here, we're not going to see the inside of the sample, we're actually going to see the outside surface. So this is the first kind of microscope that allows us to see the outside of our sample. And again, we're going to need computers to process the image here. And since we're bouncing the electrons off the outside surface, we're going to get a three-dimensional image of the outside of that object. And we're not limited by size as well, because you don't have to have a very thin sample. You can have a relatively large sample and put it inside this microscope. The way this microscope works is, if you notice in the picture here, there's like a little door. Okay, you open that door, this is what is called the imaging chamber. And whatever fits inside of that imaging chamber, we can then image with that microscope. And the price range here ranges from about $100,000 to that's $1 million plus. Okay, that's what my head is covering here. So it's $1 million or more. And really the difference in price has a lot to do with the size of the imaging chamber. How big of an object do you want to be able to scan? So like a $100,000 scanning electron microscope would have an imaging chamber maybe the size of like a jewelry box. Okay, and then $1 million microscope here might have an imaging chamber the size of like a bread box, something that you can fit like maybe a loaf of bread or something like that in. I've had people look at this image here and say, oh, could my head fit inside of that imaging chamber? Maybe, but only if you're willing to cut your head off your shoulders, because in order to image something, you have to be able to shut that door, and then it is vacuum sealed. Okay? Because we can't have air molecules floating around as we're shooting these electrons, because those air molecules could actually ricochet those electrons, which would cause a blurry image. So we got to basically close the door and suck out all the air to get a clear image. So as a result, if we were to put something alive in there, it's not going to be alive for very long. Okay? It's going to basically suffocate. The other thing that is necessary in order to get an image, electrons don't bounce off of things very easily. So in order to get them to bounce, you actually have to cover the surface that you want to image in a very, very thin microscopic layer of gold. Okay, gold basically allows those electrons to strike it and then bounce off rather than going straight through. Okay. So the images that you get from a scanning electron microscope are pretty fascinating. So here's a picture of an ant. Okay, and in this picture, the scale that you're given is 1 millimeter. So this ant in its entirety is probably about 10 to 12 millimeters long. But we can see a lot of great detail that you wouldn't be able to see underneath a normal microscope. So like you have these striations, the little stripes that seem to be following its edge. Okay, and this picture here, you can see it in a little bit better detail because it has color. Now this is again what we call false color, because electrons don't have color. Okay, all the images that come out of an electron microscope are going to be in black and white because color is a function of light. So what happens here is when the image comes to the computer can actually assign color values to certain depth. Now, it's not that this is a rainbow-colored ant. It's that we've added color to make it easier to see the details. Because, again, our eyes pick up color much easier than it picks up like a black and white image. Does anybody have an idea of what this is? It's Velcro. Very good. Okay, Velcro has a fuzzy side and then it has a prickly side. The fuzzy side are actually tiny little thread loops, and then the prickly side are little hooks, little plastic hooks, and then when you press Velcro together, the hooks get stuck in the little loops, okay, and that's what holds it together. And then when you pull the Velcro apart, the hooks bend and eventually let go of the loops on the fuzzy side. And if you have really old Velcro, okay, the fuzzy side starts to get more fuzzy. That's because the little loops start to break. And then the prickly side doesn't get as prickly because the hooks themselves can break, but they also kind of lose their flexibility. So basically the hooks and the loops don't stick together anymore. Any idea what this is? Think on the microscopic scale. This is a staple sticking through a piece of paper. Okay, paper is made out of wood pulp. Okay, so they take wood and they grind it up into tiny little fibers and they mix it in with some glue and smash it flat. So here we can see the fibers of the paper, and then the staple is the big metal thing sticking through it, but obviously it's a little tiny wire. Just here it's magnified. The like dark spots that are on the staple, that's the glue that holds the staples together inside the stapler. Okay, so you have tiny little staples glued together, and then when you smash the stapler, it bends that wire and pokes through the paper on the other side. So here's some really interesting images from a scanning electron microscope. On the left here we have a tarantula. And a tarantula is a fairly large organism, so we can see a much bigger object. Okay, and we can see some details here that you can't normally see, like it has 8 eyes on its head. You can, if you look closely at the bottom, you can see its fangs. The organism on the right-hand side, this is a type of worm, and it looks pretty terrifying, but if you were to come face to face with one of these things, you probably wouldn't be so scared of it because it's only about 1 millimeter long. So its entire body is 1 millimeter. So if you look at a ruler, the smallest marking on that ruler is the size of this thing. Its terrible looking jaws are used for eating bacteria. This type of worm lives exclusively around hydrothermal vents at the bottom of the ocean. So basically you have volcanic eruptions under the ocean, and where those volcanic eruptions happen, bacteria thrive, and these little worms swim around and eat those bacteria. It looks pretty terrifying, looks like something out of some sort of sci-fi movie, but it is in fact a tiny, tiny worm. All right, so next question. Which kind of electron microscope would be useful to see the internal structure of a virus? So let's say we wanted to see the inside of the coronavirus. Which kind of microscope would you want to use? A TEM, which is a transmitting electron microscope, a CLM, which is a compound light microscope, a SEM, which is the scanning electron microscope, or the CFM, the confocal microscope. The next kind of microscope is actually a group of microscopes known as a scanning probe microscope. And what this does is, instead of relying on shooting energy at your sample, it actually uses a microscopic probe to feel the surface of the sample. Okay? So imagine you close your eyes and you reach out and you're touching the surface of something. Okay? You can kind of get an idea of what that surface looks like by the little bumps and ridges as you run your fingers over the surface. And that's the way that this microscope works, is it takes a microscopic needle, like a very thin little point, and it runs that point over a surface. And as that point kind of bounces up and down over the surface, it sends a signal to a computer and says, this is the surface that I'm feeling. And it does this in a very specific pattern, so it gets all the details from that image. So instead of like shooting energy at the surface, we are instead feeling the surface. And these microscopes can range in price from $29,000 to about $500,000. Surprisingly, they're not as complicated to use as some of the electron microscopes. It does take a little bit more training than your typical CLM to use, but these are very commonly used in different industrial applications, because if you go into like the computer industry, whether it's making iPads or cell phones or something like that, computer chips, the circuitry now is getting so small that even typical magnifying glasses and microscopes are not able to see the size of the circuitry. So these are actually used very commonly in electronics manufacturing, because now we're dealing with circuitry that is extremely tiny. These scanning probe microscopes, depending on the type of model you use, can create extreme magnification of images, up to 1 million times magnification. Okay. So there's a couple different ways that these work. The typical one is what is called the atomic force microscope, the AFM there in the middle, where it basically has a needle, and that needle kind of bounces up and down off the surface, and it sends a signal back to the computer. You have what is called the scanning tunneling microscope, which uses that needle in much the same way, but instead of bouncing it off the surface, it uses it to focus a stream of electrons. So it's kind of like combining an electron microscope and a probe microscope. And then the one on the right here is the scanning near-field optical microscope, or SNOM for short, which I just love that name. And what this does is it kind of combines a CLM and a probe microscope, because it shines light through the sample, but that light gets funneled down a very, very thin lens, which is basically a needle-sized lens, and that focuses the light much more intently than a typical CLM or even a confocal microscope can do. So we can see some extreme magnification factors of objects underneath the scanning probe microscope. So this is a picture of human hair. A lot of us think that our hair is just like one solid strand, like a piece of fishing line or something, but it's not. Our hair is actually made of tiny little scales. And in fact, the way our hair is built, it's built out of a material called chitin, which is very similar to what your fingernails are made out of. And basically, your hair is like little scales of fingernail material kind of latched together. So if you have really long hair and you start to get split ends, that's because the scales are starting to fray apart. And then what's really amazing with some of these scanning probe microscopes like the AFM, we can actually see atoms. So this is a picture of actual atoms in a silicon crystal. Okay, now the atoms are the little balls that we're looking at, these little spheres. So we can't see the inside of the atoms, okay, but what we can see is the outer surface of those atoms. So now we can actually see the arrangements of atoms inside of molecules themselves. It's kind of amazing that we have the technology to do that now. The final thing to consider is that scientists are constantly pushing technology into new directions. So we can have what we would call the cutting edge microscopes. These are microscopes that are new ways to visualize things at a microscopic level. This is where scientists are thinking outside of the box. This is where they're thinking, well, what's another way I can look at this very, very tiny thing? Okay, what method is out there that we haven't invented yet? These are often one-of-a-kind, priceless prototypes. So these are new inventions that may only exist in one place in the world. And this is how new technology comes about, is somebody develops some brand new idea and then they have to build it on their own. So it could take millions of dollars to develop the technology. And then once that technology is developed, then we can say, okay, how can we get this technology into the hands of other scientists? So the picture that we have here, this is a microscope that was being built in the basement of a laboratory at MIT. That's Massachusetts Institute of Technology, where a bunch of very smart engineering and science nerds go to school. So this is a multi-million-dollar piece of equipment that I have no clue how it works. I just find it amusing that it's millions of dollars in research placed on top of cinder blocks. But again, this is how new technology is developed. Scientists come up with ideas and say, well, what's another way we can do this? We want to see things that are very, very small. How can we do that? And they develop new instruments to do that all the time. And this is kind of like pushing the boundary of our understanding of science. So there are still things within the realm of biology that we still haven't clearly visualized because they're just too small even for our best equipment. So talking about something like DNA, okay, we've only gotten glimpses of what DNA looks like, okay, at a microscopic level because it is so small. Okay, so there's still more that we want to be able to visualize. We just need the right equipment, the right tools to do it. Final question. See if you were paying attention. Which kind of microscope would allow for the viewing of a living specimen? There's only one kind of microscope that I mentioned that you can actually see living things under. All the other microscopes, either the way that the image is produced or the way that the sample has to be prepared, basically means that you cannot have a living specimen at the same time that you're viewing it. So is it A, SEM, that's a scanning electron microscope, B, the SPM, the scanning probe microscope, C, the CLM, the compound light microscope, or D, the TEM, the transmitting electron microscope? Which one allows us to see actual living things?